Welcome everyone. Today we are diving into one of the most important soil quality parameters, cation exchange capacity, commonly known as CEC. If you have ever wondered why some soils hold nutrients better, why certain plants thrive only in specific type of soil, or how fertilizers actually work, behind every these things, CEC is the key. Let's explore this concept in today's episode. So what exactly is CEC? Think of soil as a giant marketplace full of tiny soil particles, clay and organic matter, acting like magnets. These particles carry negative charges, and because of that, they attract and hold positively charged ions, known as cations. These cations include important plant nutrients like calcium, magnesium, potassium, and sodium. They also include some acidic cations such as ammonium, hydrogen, and aluminium. So, CEC simply tells us how many of these nutrient cations a soil can hold and exchange. Higher CEC means soil is like a nutrient bank with a large storage capacity of cations. So, higher CEC means more fertile soil. To understand CEC at work, imagine negatively charged clay particles surrounded by nutrient cations. These cations aren't glued permanently. They are weakly held by electrostatic forces, meaning they can be exchanged. Cation exchange capacity, CEC, is a measure of the soil's ability to hold positively charged ions. This soil property influences soil structure, stability, nutrient availability, and soil pH. It affects the soil's reactions to fertilizers and other additives. The clay mineral and organic matter components of soil have negatively charged sites on their surfaces. These charges attract and hold positively charged ions, cations, by electrostatic forces. CEC is measured by replacing adsorbed cations with another known example, ammonium ion or sodium ion in exchange. Look at this picture. Soil or clay particle will have negatively charged. These negatively charged attracts cations to its surface and they are held by electrostatic forces. Now there are two types of cations, acidic cations and basic cations. Base cations are calcium, magnesium, potassium and sodium, whereas acid cations are hydrogen, aluminium and ammonium. So base cations, they are known as exchangeable basicity and acid cations give exchangeable acidity. So low CEC means you have less of base cations and more of acid cations like H plus ions, aluminium ions, etc. Whereas high CEC particle will be absorbed with more of base cations such as magnesium, calcium, sodium, potassium, etc. A clay mineral or organic component of soil have negatively charged sites on the surface, which absorb and hold positively charged cations by electrostatic force. This I have already told you in the previous slide. This electrical charge is critical to the supply of nutrients to plants because many nutrients exist as cations. They act as a storehouse of nutrients for plant roots. There are five most abundant exchangeable cations in the soil, namely calcium 2 plus, magnesium, potassium, sodium, and aluminum. In general terms, soil with large quantities of negative charge are more fertile because they can retain more cations on their surface. Now, look at this picture. Plant roots will be surrounded by soil particles. Now, plant root cells convert carbon dioxide into carbonic acid, H2CO3, which then ionizes to release H plus ions. So, plant root cell surfaces will be adsorbed with H plus ions. Okay. Now, excuse, plant needs other cations as well for its normal functioning. Therefore, it will try to exchange this acidic cation H plus with other base cations such as calcium, magnesium, potassium, and sodium adsorbed to the clay particle in the close vicinity of the root cell. So, more the cations present or adsorbed onto the clay particle, more these cations will be available to absorption by the root cells of the plant. That's why a soil with high CEC will be more fertile for the plants. Now you might be wondering, how do we measure this in our lab? There are different scientific methods, but all essentially follow the same logic. First, saturate the soil with a known cation, then replace it and measure how much comes out. The common method include ammonium acetate method, sodium acetate method, barium chloride method, and summation method, which is based on individual exchangeable cations present on the surface of the soil. The most widely used is the ammonium acetate method. To give you a practical idea, let us look at ammonium acetate method, one of the standard laboratory technique. Here's the simplified workflow. Take a soil sample and saturate it with 1 molar ammonium acetate solution. You can centrifuge it to make a suspension and leave the soil suspension in 1 molar ammonium acetate solution for 24 hours. Next day, this replaces the original cations with ammonium ions. So let's repeat the step. A soil sample is mixed with 1 molar ammonium acetate solution buffered at pH 7. Now this pH 7 is really very important. I'll tell you in our later slides. This solution saturates soil's negatively charged site with ammonium ions, displacing other positively charged cations such as calcium, magnesium, sodium, and K plus into the solution. The mixture is then shaken, centrifuged, and the leachate present in that solution is displaced, cation is collected. Repeat this step multiple times to ensure all exchangeable cations are removed and collected in the leachate. So here you can see 24 hours saturated 1 molar ammonium acetate soil suspension is centrifuged and the filtrate is collected in a fresh flask. The step is repeated 3 to 4 times, maybe 5 times to remove all the exchangeable cations and collect it in the form of a leachate. Now, the concentration of each displaced cation, namely calcium, magnesium, sodium, potassium, in the collected leachate is measured using techniques like atomic absorption spectroscopy or ICP-OES. Now let us look at the formula, or how do we express CEC. The sum of measured quantities of all displaced base cations is calculated and represented in the form of potential CEC of soil at pH 7. Why pH 7? Because we used a buffered solution of 1 molar ammonium acetate at pH 7. CEC is typically expressed in the form of milliequivalents per 100 gram of soil. So this is the CGS unit, milliequivalents per 100 gram of soil. It can also be expressed in centimoles of positive charge per kilogram of soil, which is the SI unit of CEC. So CEC represents exchangeable base cations plus exchangeable acid cations. Water-based cations, calcium, magnesium, potassium, sodium, and acid cations, namely hydrogen, aluminium, and even ammonium ions. Now, what is or how is the CEC linked to soil texture? You can see here, light colored sandy soil, typical CEC would be 3 to 5 milliequivalents per 100 gram of soil. Loams will have somewhere between 10 to 15 milliequivalents. Silt loams 15 to 25. Clay loam 20 to 50, and organic soil 50 to 100. That means CEC is a good parameter to assess clay and organic soils, whereas sand and silt loams, they are very low in cationic exchange capacity. Therefore, their fertility will also be less. As I already said, units of CEC is milliequivalents per 100 gram of soil. Now, there are some important things which you must know. There are two types of CEC calculation. One is effective CEC and potential CEC. Effective CEC is actually the CEC at the native soil pH. Whatever the ambient pH of soil is, at that particular pH, if you estimate CEC, then it will be known as effective CEC. It will typically include all base cations as well as acid cations. This reflects actual charge capacity under field conditions. The second term which we commonly use is potential cation exchange capacity, or potential CEC. This is determined by extracting the soil with a buffer solution at pH 7, as we did in our method, 1 molar ammonium acetate buffered at pH 7. So what we found was potential CEC. What is this, or how this is related to acidity or basicity of soil? In alkaline and neutral soil, where the pH is above 7, the exchangeable acidity is often negligible. The CEC then becomes sum of base cations only. Whereas in acidic soil, where pH is already less than 6, H plus and Al3 plus ions, ammonium ions, also contribute significantly to the cation exchange capacity. Therefore, they must be included in the calculation. So, if the soil is naturally alkaline or neutral, then we can go with potential CEC. But if the soil is acidic in nature, naturally, the ambient pH of the soil is less than 6, then we should calculate effective CEC. So why is CEC so important? Here are the big reasons. CEC indicates soil fertility. Soils with higher CEC can store more nutrients for longer periods. CEC helps in fertilizer management, helps to decide how much fertilizer to apply, neither too little nor too much. CEC influences soil pH and nutrient availability. Soil with high CEC resists sudden changes in pH. A balanced, nutrient-rich soil leads to stronger root system, better crop yield. Understanding CEC thus helps farmers, researchers, and even home gardeners to manage soil more scientifically. Let us end with everyday application of CEC. Farmers use CEC values to choose the right crops for a soil. Soil testing labs use CEC to recommend fertilizers. Environmental scientists use it to assess pollution risk and nutrient cycling. And agronomists use CEC to classify soil and evaluate soil amendments. In simple terms, CEC helps us understand how soils behave and how they store nutrients. How best can we work with them for sustainable agriculture? So that's all about CEC. In our next video, we'll bring one more aspects related to agriculture biochemistry. Till then, keep watching our older videos on agriculture biochemistry. And if you like the videos, keep sharing and commenting to let me know your views on all these episodes. Please subscribe if you like us. That will encourage us. We'll see you in the next part of agriculture biochemistry.